Hey everyone, welcome back to the channel. If you're new, make sure to like and subscribe. In today's video, we'll be going into the second part of our microbiology series. So this has to do with microbial cell structure, mainly the cell envelope. So let's take a look at what that means. I wanted to start with an analogy here of a jelly donut. If you think of a jelly donut, you have this soft interior, which is made of jelly, and you have a donut exterior, right? And so the distinction for bacteria is the same as a jelly donut. You have this soft stuff inside, which we can call the jelly. It's actually the cytoplasm. And there's all this other stuff, all these layers outside of that, right? So if this is your jelly, this is all your donut. And so what you see, and what may confuse some people, is what the cell envelope is. The cell envelope is everything past this soft interior area. So just keep that in mind as we're going forward. You can think about it again as the jelly donut. Your jelly is your cytoplasm and your DNA in that cytoplasm, and your donut is everything outside of that. And so that should help you visualize the cell envelope versus the cytoplasm. And any cytoplasm related is going to be in the next video, not in this video. So I wanted to give you first a bird's eye view of the different types of cell envelopes. So remember how the cell envelope can have various different layers, right? And we'll get into that, but I just wanted to give you this bird's eye view first. So if we start with the gram-positive bacteria, they have a cell wall and an inner membrane. Now, let's contrast that with the gram-negative. They have an outer membrane, a cell wall, and an inner membrane. So we'll go back to the gram-positive. I just wanted to show you that your cell envelope can have many layers or less layers or more layers. In the gram-positive, you have this thick peptidoglycan cell wall. The purpose of this is so that your cell doesn't burst, right? That's the main thing, is if your cell gets too big, it'll be like a balloon and it'll pop. So you have this rigid cell wall that keeps it from popping every time it divides. Now, it's made of two parts, right? You have this chain of sugars, as you can see. This is a sugar. And you have these peptides coming off, these little circular things. And so let's get into that. The first part, these sugars, is the backbone. As you can see, it is just a bunch of layers of sugars like this. Now, the way these sugars are connected are by these peptides, right? And you can see that connection right there. And so we need to talk about both parts. So we'll start with the backbone. The backbone, as you can see over here, is made of two sugars, what we call the NAM and the NAG. Now, if you want the actual names, they're right over here: N-acetylglucosamine acid and N-acetylmuramic acid. Now, you don't have to worry too much about the names. If you're really curious, N-acetyl is just a group that's added. It's a functional group in organic chemistry. And glucosamine acid is just the sugar that you have. So, same thing with the muramic acid. It just has an N-acetyl group on it. Now, these are slightly different. The reason for that is because your NAG and your NAM need to do different things within the cell wall structure. So, again, they form the backbone. And so it begs the question, what is the difference between NAM and NAG? Why do we need two different sugars? The purpose of the NAM over here, as I've drawn for you, can basically attach peptides onto it. Now, for reference, I'm not sure which of these sugars, like which carbon is going to bind the peptides. That would be something that is above the course that we're in currently. But I'm just going to draw it on carbon 2 just so you have an idea that it binds like that. So just remember, this is just an example. If you go somewhere else and you say, hey, it was actually on carbon 5 or it was actually on carbon 3, this is just to give you an example of what it looks like. It's not the actual position of it in 3D space. So as you can see on carbon 2, you can have these peptides on the NAM. And so the reason we want the NAM is to allow for that peptide cross-linking, which is this part right here. We need this to connect the two layers. Now then, what's the point of this nag over here? The nag is basically like a spacer, right? You need some space in between where you aren't adding peptides, because you don't want everything to be completely touching. You still need some flexibility in that cell wall. Otherwise, it's basically an actual brick wall, which is not something we want. Now, let's look at what this actually looks like. Over here, I've drawn it out for you. There's a lot going on, so we'll start on the left, the very top. Look at this. You have this right here is your backbone. Again, this right here is your backbone. This is row 1 and this is row 2. So if you need reference one more time, this would be row 1 and this is row 2. The first thing I want you to look at is that row 1 goes left to right. Row 2 goes right to left. They go in opposite directions, what we call anti-parallel. So the peptides that we talked about earlier and we didn't get into are amino acids. So amino acids over here, it just says it's a polypeptide. So you have a long chain of individual amino acids on there. And that's what this is. So let's take a look at it. I've drawn it out here on the side for you. Let's remove this just so you understand what's going on. So you have your NAM sugar over here, and coming off of it, basically hanging off, dangling off, is a bunch of amino acids, a polypeptide chain. And you have your second row, which is another NAM down here, and it has these things coming off of it as well, which are the amino acids. Now, just think about this. If you have these things and they're kind of separate, and you want a strong cell wall, you don't want them to be just floating around. Then you don't have a wall. You just have a bunch of layers next to each other. And so what happens is these guys need to get close to each other, and obviously they need some space, but they still need to be close together. That's why one goes this way and the other goes that way. So the way these guys bind together is through the penicillin binding protein. So before we talk about what this does, let's look back over here just so you can see it with the amino acids. This is your chain. You have this L-lysine over here, and it has this 5-glycine bridge. You don't have to know exactly what these are or what they look like. Just know these are amino acids and they're connected to each other. And so the reason for this is you need something to bring these two closer together. And so as you can see here, the 5-glycine bridge would look something like that, right? It brings these two guys closer together. Now, there's an enzyme that does this, but we don't have to talk about it. You don't have to worry about it. All you have to know is that when you're forming the cell wall, this 5-glycine bridge is there, and it's close enough for these two to start to bind together. Now, the way they actually bind together is through the penicillin binding protein. So as you can see in purple, what that does is it removes this D-alanine amino acid, cuts it off, and then it forms a bond between this 5-glycine bridge and this D-alanine of the second row. And so what that is, it does this, right? Connects these guys together, and it cuts off this guy over here. And now these guys are formed together. You have 2 rows of a cell wall that are actually linked now. And that's what this looks like over here, this peptide cross-linking between the sugar backbones. Now, one thing we didn't talk about, and I wanted to bring it up right now, is how can we mess up the cell wall, right? Because we've talked about how the cell wall is formed. So what causes issues? The first thing is these 2 sugar backbones. This is the first part that we can mess up. So you have this beta 1,4 bond that I hinted at earlier, right? This keeps that sugar backbone together. Now, what if we were to cut this open, right? We were able to cut this bond, the backbone would fall apart, right? You wouldn't have a cell wall anymore. Now, what we've seen is that there's this enzyme called a lysozyme, and it's able to cut the beta 1,4 bond. Now, beta 1,4, all that means is the 1 carbon on the left or the right of the NAG and the NAM. As you can see over here, there's 1 carbon and there's 4 carbon. So that doesn't mean number of carbons, it means the number of that carbon on the individual sugar. So as you can see over here, this is the 1 carbon, this is the 2, 3, 4, 5. Same thing over here, 1, 2, 3, 4, 5. What happens is you form this bond between the 1 and the 4 of the NAG and the NAM. And lysozyme can come in and it can cut it, and that disrupts the entire cell wall. Now that we understand how to break the backbone, we have to think about the peptides, right? Let's say the backbone is just fine, right? What else could happen? As I've hinted over here, we have penicillin, which is an antibiotic drug that we take when we're sick if you have a bacterial infection. What that does is it basically binds to the penicillin binding protein. So instead of penicillin binding protein cutting this off and binding these two things together, penicillin will basically join in and it'll stop the penicillin binding protein from even coming over here. So what that does is it disrupts the process, so this guy doesn't get cut off, this guy doesn't bind anymore, and you make the cell wall a lot weaker. It's no longer connected, it's back to this. So this is way too far to actually do anything, and so it's not bind together. Now, the other way, now that we understand how to break up the cell wall in 2 different ways, these are issues that bacteria have to deal with. And so how they do that is they also have what we call teichoic acids. What these do is they strengthen the cell wall even more, so that if anything happens, these are still in place, sort of like a safety measure. And so what they do is the teichoic acid, just teichoic acid, will strengthen the cell wall and it'll help with adhesion to surfaces. Now the lipoteichoic acid will connect the cell wall to the cell membrane, which is the layer below it in the cell envelope. Now, the cell membrane stuff is all over here, but we won't touch on it until we've gone through the other types of cell envelopes. The reason for that is because these 4 share the same cell membrane structure. So let's go to the gram-negative bacteria. Now, the gram-negative bacteria have an outer membrane, a cell wall, and an inner membrane. So I've drawn that out for you over here. So let's take a look at the outer membrane first and then work our way in. Within the outer membrane, this is what it looks like. This is the whole structure, and we'll break it down. We have the top and the bottom. So we're going to start at the very top and work our way in, like I said. The function of the outer membrane is selective permeability, so only allow certain things in, protection against basically anything that's outside that wants to hurt the cell, and cell rigidity. So kind of the same thing, stop the cell from bursting every time it wants to reproduce. So right now we have the first part is the lipopolysaccharide. And so what that is it's this entire layer over here from this side to the bottom. And it's broken up into multiple pieces. As you can see, this is the fatty acids, and then everything else is kind of blurrier in this picture, but we'll break it down. First thing you have is this O antigen. What that is a chain of sugars. And as you could tell from the name, O is outside, so it's the most outer thing in the cell membrane, in the cell envelope, and it's an antigen. Antigen just means if it's in any other organism, the immune system will react to it. So usually antibodies, which are proteins, will bind to it, and they'll either have a good reaction, no reaction, or a bad reaction. Now, one thing to keep in mind is it's a virulence factor. So why it's a virulence factor is because it's different per strain. So if you think about a family of E. coli and you have a bunch of different members of that family, each of them have an entire lineage beneath them, right? And so what that means is you get a whole lineage of similar E. coli, but they're not identical. So these guys are identical because they're part of the same family, but these guys are not. And so what you see is that we call each of these strains, as you can see there's 3 strains here, that are all E. coli, but they're slightly different. One of those differences that allows us to call them different strains is this O antigen part. this is different per strain, and it's one of the ways that we can tell that this is slightly different than the other strain of E. coli, but it's still E. coli. Now, after that, going down is the core polysaccharide. So, again, another chain of sugars. Sugars are everywhere, and it's constant across strains. And what it does is it connects the O antigen on the top to the lipid A on the bottom. So that's literally all it does. It's the same across all three strains that we've shown over here, and just strains in general of the same family. Now, lipid A is a phosphorylated sugar backbone. So what does that mean? It means you have a sugar, as you can see over here. Same idea as before, it's a backbone of sugars, so it's a chain that goes side to side, and it's filled with sugars. Now, the phosphorylated part just means it has this phosphate group on the side, as you can see there. Now, you also have these acetogenic fatty acids. So, this will come up again in the cell membrane, so just keep that word in mind, but don't focus too much on it right now. Now, there's also the fact that it's an endotoxin, and so again, we talked about how it's not safe to have these bacteria. They cause infections and stuff. One of those things is that it's an endotoxin. It'll cause fever or sepsis in organisms. Now, the other part of this membrane, this outer membrane, is the porins and the membrane proteins. So as you can see here, porins are hydrophilic passageways. I drew a little diagram for you over here that it just means it loves water, right? So what that means is things that are hydrophilic can go in very easily, if they're small enough, through this porin and make its way into the cell. So that's what that means. And hydrophilic just means water-loving. So they'd have to be around 600 to 700 Daltons is kind of that small enough size. Now, there's also the membrane proteins. These allow for active transport, which means things that aren't small and hydrophilic being allowed to pass through selectively. Structural support, it helps to keep this membrane together, and they increase virulence, as we've talked about before. So, now that we've gone through the outer membrane, we can go back into the cell wall. So the cell wall you'll find is very similar to the cell wall of the gram-positive. However, it's much smaller, and there are some differences. So, first thing, it's a much thinner layer of peptidoglycan. Same structural thing, it stops the cell from popping. But what we see is that we do still use the NAM and the NAG. Everything here is the same, the lysozyme can still cut it up, and so that part is the same. The difference is in the binding of these together. So the peptides change, the amino acids are slightly different. What you see is now you have a DAP instead of what we had over here was an L-lysine, right? So we had a lysine with a 5-glycine bridge. Now we have this thing called a DAP and D-alanine. And so the same thing though, the penicillin binding protein will come in, it'll cut this, and it'll bond the two together. But because of this amino acid's character, which you don't have to think about too much, it doesn't need a glycine bridge, and it forms a bond directly between the DAP and the D-alanine. So you can imagine it's much closer together. Whereas over here, there's an entire 5-glycine bridge holding these guys together. And so that's what the cell wall looks like here. Now, the next thing would be Mycobacteria. These are gram-resistant. They don't get gram stains at all. So don't use that classification for Mycobacteria. They have an outer layer, not necessarily a membrane. They have a cell wall, and they have an inner membrane. So let's get into that. This is what Mycobacteria's outer layer looks like. First thing to keep in mind, it's very hydrophobic. It does not like water. Now, don't worry about these little sugars over here that I've highlighted down here. It says don't worry about it. So those you don't have to worry about. But what you see is that it has porins again. That same concept that we just talked about earlier. It allows small hydrophilic molecules to pass through. So water-loving molecules or water itself can pass through this porin. And so remember, everything outside here is very hydrophobic. It's basically oil or wax. Does not like water. And so water has to sort of sneak its way in through these porins, and so these guys grow very slowly. It's very hard to get water in there. Now, the second part, once you get through this, is the cell wall. So, wouldn't you believe it? It has the same thing as the gram-negative. It's a tiny, thin layer of peptidoglycan. It does the same thing. It stops the cell from exploding. Again, it can be cut by the lysozyme. It has the same sugar. And in this case, it actually shares the same AP-D-alanine linkage as the gram-negative. And the penicillin binding protein will go in, it'll cut this, and it'll bond the two together. And no glycine bridge. So, if you think about it, the cell wall is very similar to the gram-negative bacteria. But the outer layer is very different than the outer membrane of the gram-negative. So, now that we've gone through that, we can talk about the mycoplasma. These are bacteria with no cell wall. As you can see, they just have a single membrane. So it's not really stable on its own. Remember how we have these rigid things called the cell wall, keeping the cell from popping? These guys don't have that. They usually, to combat that, will live within other cells or organisms. And so what that means is they're kind of like parasites. They're intracellular parasites. So they will live in other cells or in other bacteria because they can't necessarily provide for themselves. The one thing that's interesting about them, and we'll talk about what hopanoids are when we get into the cell membrane, is that they can't produce their own hopanoids, right? So they steal from the eukaryotes. They're usually in eukaryotes, so this will make sense when we go over the cell membrane. And if you're confused, I will loop back around to this. So for all of that to make sense, now we've passed all the layers of the cell envelope. We're at the last layer of the cell envelope, right? Which is the cell membrane. So the reason I did it like this, again, is all four of these share the same type of cell membrane. So let's start with the functions of the cell membrane. It acts as an osmotic barrier. What that means is ions, so positive or negatively charged things, can't just randomly pass through. It creates a proton gradient. And so what that means is, since you're only allowing some positive charge to go through, some negative charge to go through, you can regulate it, right? And when you do regulate it, you create this gradient where there's a little bit of plus charges over here and a lot of plus charges over here, and you can change how that is, and you can use that as the cell for different things, right? The other thing it can do is help with transport. So there are proteins on here that I haven't drawn out for you, but there's proteins along this wall, which are over here. As you can see, this protein seems to let things right in. This one is structural, etc. There's all these different types of proteins that allow stuff in, and that's what this is talking about, is active transport of certain things. So you can imagine you as a cell don't want certain things inside. So you have these openings that only allow certain molecules into it. At the same time, only certain things can pass through easily. Some things require more work, etc. And so what we can see is that carbon, nitrogen, phosphorus, sulfur, vitamins, these things can't pass through super easily. Whereas passively, water, gases like carbon dioxide, nitrogen, these kinds of things like oxygen as well, can pass through quite easily. They can just diffuse through. They go through this area right here. Now, the other thing is energy generation via the electron transport chain, which is something we sort of touched on in previous biology classes, but you don't have to worry about necessarily for right now. The other thing would be signaling, which again, you don't have to worry about, but it is one of the functions that is done by the cell membrane. Now, I did talk earlier about this idea of an acetogenic lipid earlier in the video. That's basically just this right here, this fatty acid. So acetogenic just means it's made of acetyl groups or acetate groups, right? And so let me show you what that looks like. So as you can see, this is your acetogenic lipid. So all you have to know is that that's what it's called, right? You don't have to worry about the specifics of this. It's just called an acetogenic lipid, and it usually looks like this straight chain. That's all you have to worry about there. Now, the other thing that you have to worry about is the ester linkage. So what happens is you have this acetogenic lipid, and it's bound to this thing called a glycerol. The glycerol is in the middle over here, and your phosphate. So remember, these are called phospholipids. So this is your lipid, this is your phosphate, and then you have this thing in the middle called a glycerol, which all it does is it connects the acetogenic lipid and the phosphate group. So let's show you over here. This blue thing right here is your phosphate, this is your acetogenic lipid, and this is your glycerol. It helps to connect the two, right? And so the way the lipid is connected to the glycerol is through an ester linkage. And this will become important later on. You don't have to worry about what an ester is. It is a part of organic chemistry. But all you have to know is that it uses an ester linkage. So just remember, acetogenic lipid, ester linkage. That's it. Now, the other thing is that it's a phospholipid bilayer, bi meaning two. So as you can see, there's a space in the middle. And if you imagine this line right here as a mirror, everything on the top is the same as the bottom. These are two different layers of phospholipids. And as you can see over here, and this is your hydrophilic region. So this loves water, this does not like water. That's why it kind of forms this bilayer structure. Now, in terms of the cell membrane, the rigidity of it, so how strong or smooth it is, it can change, right? As you can see over here, this looks pretty straight. Whereas over here, it's curvy and it's kind of different than you would imagine. And so the way this happens is through hopanoids or sterols. And so what these are, a sterol is basically like cholesterol. Hopanoids, I don't have a particular example of, but they're what bacteria use. So the reason we need this is because you want this cell membrane to be flowy and you want to be able to change how thick it is. So if you think about jelly, right? Some people like their jelly super runny, whereas others like it much harder, right? And so that's the idea here, is you as the cell want to be able to control how fluid your cell membrane is. And so you can use hopanoids if you're a bacteria, or sterols from other eukaryotes. And so let's go back over here. These guys, the Mycoplasma, are the only ones that can't produce their own hopanoids, and so they steal sterols from eukaryotes to change the rigidity of their cell membrane. Whereas everyone else over here in the bacteria, gram-positive, negative, and Mycobacteria, form their own hopanoids, and so they have easier time changing their fluidity like that. Now, the other thing on here is these integral membrane proteins that we talked about. So like I told you, they can do transport, signal reception. They can either help hold the entire membrane together, or they can sort of help with movement in a sense. So now that you understand, we went from the top all the way to the bottom. And so what I want to do is talk about Archea, and then we'll get back into accessory structures. So just keep following along so you're not confused. Now, Archea are their own separate domain, not in the same domain as bacteria. So what they have in their cell envelope is a cell wall or an S layer. And we haven't talked about what an S layer is, but just keep that in mind. And they have an inner membrane, which is different than this inner membrane over here. So let's start with their cell wall. They use what we call pseudomurin or pseudopeptidoglycan. So it's the same idea as peptidoglycan, but it's sort of pseudo meaning fake, right? It's fake peptidoglycan. It's not necessarily the same thing. So they have that same idea of the repeating sugar backbone. And they have, instead of NAM and NAG, they have NAG and NAT with a T. And so this is slightly different, but there's still these sugars. And the other thing that's different is instead of having a beta 1,4 linkage, now they have a beta 1,3 linkage. And so let's talk about what NAT is, right? Because we haven't talked about it. NAT allows for peptide cross-linking. So it's the same as before, except now it's a slightly different sugar. And so, same idea, you have this peptide cross-linking between NAT and the NAT down here of the second layer. And so what you see is that they are bound together. There's no 5-glycine bridge or anything. And so it's different than in the bacterial cells. So remember how we talked about we want to destroy this cell for some reason, right? We can't do that with the lysozyme anymore. This only breaks 1,4-beta bonds, and so you can't break this using a lysozyme, which makes this even better at infecting things because we can't use this to kill it anymore. And we won't go into exactly how this is formed over here because it's not a penicillin binding protein. It's something similar, but it's an archaeal enzyme, and we don't talk as much about archaea in this class. But it's the same idea of this bond forming and creating that 2 different layers of sugars. One goes left to right, the other goes right to left. Now, the other thing that you could have as an archaea if you don't have this cell wall is what we call an S layer. This is a layer of proteins or glycoproteins. So you can see over here, it looks like this kind of mesh, and it would replace the cell wall as archaea. The functions of it are to protect or structural. So in terms of protection, it'll stop phagocytosis. So if you're a bigger cell and you have this microbe over here, you're the archaea, in this case the blue, another cell can come in and eat the archaea because it's so small. So the S layer protects from that. The other thing is you can have phage infection. So the phage over here is just a virus. And as you can see, it's kind of like pooping out DNA or RNA, depending on the virus, into this microbial cell, which is an archaea. And so now, if you have this S layer, it can't do that. So this is not possible. It also stops desiccation. So if you won't get dried out. So you'll still have retained water because of this S layer, and it'll help with metal toxicity. So if there's a bunch of metals around, it won't necessarily kill you. The other thing is, again, osmotic pressure and keep it from popping. So it's the same idea. It's always this outer layer or cell wall is to keep this bacteria from popping like a balloon. So now that we've gone into that, let's go into the cell membrane, right? The cell membrane is the inner part. As we said, we always start from the top and we go down. The functions are, again, osmotic barrier, so ions can't just pass through willy-nilly, and you form that proton gradient. The other thing is, again, carbon, nitrogen, phosphorus, sulfur, vitamins. They have a hard time passing through, so they need certain active transport proteins. And then water and gases can pass super easily. Same idea, energy generation via the electron transport chain, although it's slightly different than in bacteria. It'll use slightly different things. That's why I didn't go too much in detail on it. And the same idea of the signaling. Now, archaea can have a lipid bilayer, which we saw earlier, or they can have a monolayer. So look at the difference between the two. If you draw a mirror, this is the same as this. Over here, you can't do that. It's all one piece connected from both ends. And so what they use is sort of that same idea. There's a phosphate, there's the glycerol, and there's the fatty acid. And so let's take a look over here. Now, remember how we talked about acetogenic lipids? These are not acetogenic lipids. These are isoprenoid lipids. So let me write that for you. Isoprenoid lipids. You don't have to know exactly what that means. Just know that it is an isoprenoid lipid. You can see the isoprene unit over here, but you don't have to worry about it. So this right here is our fatty acid. This is now an ether linkage, not an ester, an ether linkage. And as you can imagine, the glycerol is over here. And if you don't remember what the glycerol was, it's right here, this green part that connects the phosphate to the fatty acid. Now they're connected by an ether. The other thing you can have, like we said, is a lipid monolayer. And so you can have the phosphate here, the glycerol here, and the fatty acid just going all the way down and then connected again to another glycerol and to a phosphate. So one thing to keep in mind as to why you would choose a monolayer as Archea, it's much better at handling extreme temperatures. So just keep that in mind. Now, as you can see, it's a longer chain and it's connected on both ends by this ether linkage. Now, I wrote that out for you over here. And one thing to keep in mind is it can be a phospholipid or it could just be any other group. So when I talk about this blue portion, I always say phosphate, right? In terms of Archea, it could be a phosphate or it could be another group. So just keep that in mind. It doesn't have to be a phospholipid. Now, again, you as a cell want an ability to change how thick your membrane is or how fluid it is, right? And so now Archea use what we call terpenoids. So we heard about hopenoids, sterols. These guys use their own thing called a terpenoid. So just keep that in mind. You can change the rigidity of Archea by using terpenoids. So now that we've gone through the things that bacteria and archaeal cells have, we can talk about some accessory structures. So let's go back to the donut analogy. As you can see over here, this donut happens to have powder on it. It could just be a glazed jelly donut. It could be a powdered glazed jelly donut, right? So you can add things to it. You can go crazy and add sprinkles to your donut. I don't know why you would do that on a jelly donut, but you can. That's the whole idea. So some accessory structures. These normally appear in bacteria. Again, we don't talk about Archea that much, so I won't get into the specifics of that, but let's focus on bacteria. They can have a capsule or slime layer. The reason it's capsule or slime layer is because it's technically the same thing. Let's break it down. This layer is a high molecular weight polysaccharide. So think about a bacterial cell spitting out a bunch of sugars. That's what high molecular weight polysaccharides are. It just spits out a bunch of sugars. What does that help with? It helps with desiccation, phagocytosis, and adherence to surfaces. So remember what we said. Desiccation means it won't dry out, it'll retain water. Phagocytosis, you won't have a virus coming in and pooping its DNA into the cell. And adherence to surfaces helps it stick to stuff. So a capsule and a slime layer are the same thing. So how would they differ? The capsule, why we call it a capsule, is it has a mechanism to attach to the cell. So it sticks to the cell. The reason we know this is because when a cell moves, it retains the capsule. Versus a slime layer does not stick to the cell. So you can imagine the sugars are connected to each other, right? But when the bacteria moves, it moves by itself. So you end up with bacteria and the sugars are left behind. So that's why we have the slime layer or the capsule. You can't have both because the second it's attached to the cell, it's a capsule. The second it's not attached to a cell, it's a slime layer. The other thing that you could have is what we call appendages, right? So our appendages would be our arms or our feet that allow us to move. Bacteria, and technically Archea, but we won't get into Archea, have surface appendages. So remember how we talked about this jelly donut, right? If you have your powder or whatever it is, these are things that are sprinkled on top of the cell envelope, right? And they are what we call appendages. So the first thing is the flagella. So as you can see in this diagram, you can have it with one flagellum, so just one of these guys. You can have a ton of them. You can have way more of them, but they fall into 4 categories. If you have one, it could be at one pole, or it could be at the other pole, right? Same idea. The other thing is you can have more than 2, and you can have it again at one pole, or you can have it at both poles. The third type is where it's everywhere, like this, where you literally just have flagella everywhere. Now, a special case is the spirochete. It has it in the periplasm. And so if we go back to what the periplasm is, as you can see, especially in gram-negative bacteria, you have the cell wall, right? And you have this right here, this blue part is your periplasm. Your cell wall is within this sort of layer of, I don't want to call it jelly, but that's pretty much what it is, some kind of gelatinous space, right? And so if you're a spirochete, you have your flagella within that periplasm. Now, what's the point of having a flagella? It helps with swimming motility, aids in adherence and in movement. So if you're familiar with the term motility, you might be asking, doesn't motility mean movement? Yes, it does. But this is specifically swimming movement, right? So if you're a bacteria within some kind of liquid media, you're able to move around using your flagella. Now, it can also aid in adherence. So you can imagine if you're on a surface, you can use your flagella to stick to that surface. The other thing is it can move, right? Whether that's swimming or on a hard surface. That's why we call this swimming motility versus movement. Now, the way it moves is through rotation. So you can see over here, this looks kind of crazy, but this whole thing right here is a motor. That's all you have to worry about. It's a motor, basically. It's what we call a basal body, and it's just a molecular motor. All it does is it spins. So it's just spinning, right? And it's powered by the proton motor force. So as you can imagine, this is a motor, so it's run by the proton motor force, right? And it's based on that proton gradient, which we talked about earlier. Because you're able to keep certain charges out and put them in, you form this gradient. And so it causes the flagellum to literally rotate. So you can imagine having a long piece of spaghetti that you're just moving around and you're making it spin, and that allows you to move. Now, how the flagella is put together looks like this. You don't have to worry about every single step, but what you should know is that this motor is created first, and then once the motor is created, you add this hook piece, and from there, the most important part that you know is that it's formed from the bottom right here. It goes through this whole thing, and it gets added to the top. So what you want to think about is the tip is growing, right? You're taking these pieces and you're adding them to the top. You're not adding over here to the base and pushing that out. You're adding to the tip. So just keep that in mind. And it self-assembles. Once you have this motor, it'll just self-assemble the entire flagellum. And so what you have is also another thing, which is called the pili / so as you can see, you can have pili / fimbriae. Now you can have both flagella and pili. So let's get into the types of pili. There's a sex pilus, which is basic allowing the transfer of material to other cells. As you can see over here, this long thing. You can have a type 4 pilus, which creates motility or movement by twitching, and we'll get into how that works. Or you can have fimbriae, which help with adhesion to surfaces. Now, let's talk about how these guys form. They use molecular chaperones, which just means they need help, right? And we don't have to get into exactly what chaperones and stuff are helping them, but they need help. They can't self-polymerize. And so these guys add to the base and they extend out. So as you can imagine, with the flagella, you're adding to the tip, and that's growing. With these guys, this part isn't hollow. And so you're literally adding to the base. As you can see over here, you add to the base and it extends up. So you're pushing the base and you're increasing the base. So as you can see, I wanted to show you this part. Polymerization is when you're adding and the chain is growing. Depolymerization, you're basically removing again. So you're taking off pieces. And so if you can imagine, if you have this long chain attached to something, right? And you're a bacteria. You've attached yourself using your pili to this thing over here. Then what you want to do is you want to start shortening that distance. So you take out this one. Now, as bacteria, you've gotten over here. So the way you've moved is by polymerizing. So you form this and then depolymerizing. You take these off. And this is actually powered by ATP, not by a motor force. Now, the way to distinguish between flagella and pili is their size. So look at this. This right here is a cell. Cells are about 1 micron long. flagellum are 5 to 10 microns long. So they are very, very long. So that's usually how you're able to tell. Now, these guys, the pili or fimbriae, are way smaller. As you can see, they fit onto the same picture as the cell. Even though it looks very long, it fits into the picture with the cell, right? Whereas over here, they're so long, they don't fit on the same picture. So just keep that in mind. Hopefully this has been helpful. Again, make sure to like and subscribe, and I'll see you in the next